This is a part second of somatic embryogenesis. Now we are going to visualize a slide regarding somatic embryogenesis. Okay. Now here we'll look for the introduction. Plant embryogenesis is the process that produces an embryo from the zygote by asymmetric cell division and differentiation of undifferentiated cells into mature tissue and organs. Okay, in general, what is plant embryogenesis is explained in this technique. Okay, now in somatic embryogenesis, LILSU, a process where an embryo is derived from a single somatic cell or group of somatic cells. Okay, somatic embryos are formed from the plant cells that are not normally involved in embryo formation. So we know that leaf cell, stem cell, root cell, these are not cells which are involved in the formation of embryo. The involved cells are pollen grains and ovules. Okay, so when these cells not involved cell form somatic embryos, okay, that is somatic embryogenesis. Okay. These embryos which are generated from somatic embryogenesis are called as embryoids. Remember that this definition can be asked in examination. What is embryoids? Then you should write the embryos which are formed by the process of somatic embryogenesis is known as a embryoids. Okay, it was very first the process was discovered in carrot tissue, okay, by Stewart and Reinert. The most basic requirement for the embryo development is physical and chemical environment, which is available only inside a magic bath of embryo sac. In nature, the embryo formation occurs only in the embryo sac. In vitro embryo can be developed if you provide the nutritional condition as in the magic bath. So magic bath is what we say, it's not a scientific word here it is used, but okay, but still magic bath is nothing but the exact required exact composition of nutrients, hormones that is required for the formation of somatic embryo. Now here we can see the different stages of somatic embryo. We can stage the different stages of somatic embryo. Okay, I have already distributed you the notes, and in notes this diagram is present. Okay, now which factors affect somatic embryogenesis? Number one is the explant. Number one factor is a explant. Explant as a source material to induce somatic embryos are very diverse. There are very responsive plants such as carrot, in which any part of plant can be used to induce somatic embryo. Means there are few plants like carrots of which any plant part is capable of inducing somatic embryos. There are some very recalcitrant plants such as cereals and legumes in which explant varies even with the genetical identity of species. So explant selection, which type of plant you are using for creating somatic embryogenesis is very most important. Now there is various types of explant: immature zygotic embryo, inflorescence, cell suspension culture, petioles, protoplast, leaves, stems and root. So all these 8 different types of plants part can be used for the generation of somatic embryos. Now plant growth regulator. So auxin is the most important. 2,4-D has been the best synthetic auxin used for inducing somatic embryogenesis. So this may be a question for fill in the blanks. Which auxin is responsible best for the formation of somatic embryo? Then your answer should be 2,4-D. Continuous supply of auxin causes embryogenic cells to divide without the appearance of embryos. Okay, so continuous auxin supplies divide these embryogenic cells. Okay, Witherell has suggested that continuous supply of auxin induces endogenous ethylene production which suppresses the embryo development. So embryogenic cells after treatment with auxin must be transferred to auxin-free medium that constitute the embryo development medium. So auxin is initially responsible for the development or formation of callus, and then these cells need to be transferred into auxin-free medium. Then there is a cytokinin. Cytokinin produces globular embryo from initial embryos. So cytokinin is responsible for what we say maturation of embryo. Okay, zeatin is promotive when applied to embryogenic cells after 3 to 4 transfer from proliferation medium to elongation, what we are maturation medium, where BAP and kinetin have inhibitory effect on embryogenesis. So zeatin is generally very less used. Okay, high ratio of cytokinin than auxin induces shoot formation and reverse favor rooting formation. So this must be also avoided. Gibberellins means gibberellic acid inhibits somatic embryogenesis. Abscisic acid promote embryo maturation and prevents germination and secondary embryogenesis. Okay, so abscisic acid is also responsible for the maturation. Now there is a nitrogen source. Reduced form of nitrogen is sole source of embryo formation. So you need to use reduced form of nitrogen. Now polyamines involved in vitro and in vivo somatic embryogenesis, involved in cell growth, proliferation and aging, interact with negatively charged molecules such as DNA, RNA, and also polyamines such like putrescine, spermidine, spermine. Okay, these are shown to drastic increase in somatic embryogenesis. So this is also a factor, that is polyamines. Then there is a genotype. Genotypic effect of somatic embryogenesis occurs as for regeneration by Schubert differentiation, and this genotypic effect is very vast. Then there is electric stimulation. Exposure of explant to mild electric current also promoted the embryogenesis. It has been found that it may promote the embryogenesis or differentiation of organs. Now this is very critical. What are the stages of somatic embryogenesis? So induction, and auxin specifically 2,4-D is generally necessary for the induction of embryogenesis. Already we have discussed that auxin induces somatic embryogenesis. Then requirement of exogenous auxins for induction of somatic embryogenesis depend on nature of explant. So concentration of auxin is solely depend on which type of explant we are using. We have already seen with different type of explant we can use for somatic embryogenesis. Now development. After reinitiation of cell division and period of cell proliferation in presence of auxin, embryogenic cells are released into auxin-free medium. So now the next step is to transfer these cells into auxin-free medium. Okay, mostly the composition of medium may be same, but there is only removal of auxin. These cells are in cluster of cytoplasmic cells called as proembryonic mass of cells. The definition can be used. Okay, so in case of development, those cells which are going to form somatic embryos are known as proembryonic mass of cells, that is PEMs. Then their next stage is a maturation. The quality of somatic embryo with regard to their germination or conversion into plant is very poor. This is because the apparently normal looking somatic embryos are actually incomplete in their development. Okay, so the normal somatic embryo that we look are not fully developed, hence their germination and conversion to plant is very low. Hence, these somatic embryo which are formed inside a in vitro culture needs a maturation medium. So it needs to be mature because it should acquire enough amount of nutrients, proteins, okay, so that they can tolerate the desiccation and harsh environmental condition. So abscisic acid has been found very essential for the maturation of embryo. Now there is a type of somatic embryogenesis. There are two types. You can refer the notes: direct somatic embryo and indirect somatic embryo. So when embryos are formed directly from the explant tissue, creating the identical clone without producing intervening callus. So direct somatic embryogenesis involves with no production of callus. The explant which are capable of direct embryogenesis seem to carry pre-embryonic determined cells. These cells are committed to embryo development and need only be released. Then there is an indirect embryogenesis which happens through the callus. Here induced embryonic determined cells are. So what happens in case of direct somatic embryogenesis, there are few cells inside the explant which are already destined to be embryo. But in case of indirect somatic embryogenesis, we have to induce some cells to be the part of somatic embryo. So here is a diagram of somatic embryos. Okay, now indirect somatic embryos. Now there is a differences between the zygotic embryo and somatic embryo. So somatic embryos are formed by sporophytic cells, that is diploid cells, whereas zygotic embryo is formed by the gametic cells, that is anther and ovule. Pollen and ovule. Somatic embryos store less amount of embryo specific reserve, whereas seed storage protein, carbohydrate are the characteristic feature. Because what happens in case of zygotic embryo, it forms a seed coat, and seed coat has a lot of nutrients which is not present in the somatic embryo. A distinct suspensor is absent in somatic embryogenesis. Even if it is present, it may not be functional as in a seed embryo. A well-developed distinct suspension is present in zygote. Suspension is a structure, okay, which is absent in somatic embryo and present in the zygotic embryo. Now, embryos have no vascular connection with the cultured explant. Definitely, somatic embryos have no vascular connection with the explant, whereas embryo has a vascular connection in explant zygotic embryo. Somatic embryos are generally lack a dormant phase and often show secondary embryogenesis and okay. But what happens in nature? Zygotic embryo do not show secondary embryogenesis and any kind of cotyledonary activity. Somatic embryogenesis has a high rate of propagation, whereas there is a low rate of propagation in zygotic embryo. Now, advantage and disadvantage. So, advantages, it is observed as it is various culture conditions can be controlled. So, it is under control. We can control the conditions and observe its development. So lack of material is not feeling the limiting factor for experiment. There is lot amount of material available. So high propagation rate, somoclonal variations can be studied, germ plasm conservation can be done. It is labor saving and eliminate disease and viruses. If we look at the disadvantages, it is confined to very few species, and it has very poor germination, okay, and it is unstable for long culture conditions. Okay, this is how we end the somatic embryogenesis lecture. Thank you.